The overall goal of this procedure is to design and use Chitosan interfering RNA nanoparticles for gene silencing studies in mosquito larvae. This is accomplished by first preparing Chitosan interfering RNA nanoparticles by the combining of Chitosan and interfering RNA, followed by heating, vortexing, and centrifugation. The next goal is to mix the nanoparticles with larval food and agarose to prepare food and nanoparticle-containing gel pellets. The nanoparticle food is fed to the mosquito larvae, and larvae feeding on the pellet is observed. Ultimately, following verification of gene silencing by quantitative real-time polymerase chain reaction or in situ hybridization assays, loss-of-function phenotypes can be examined.
This method can help answer key questions in the vector mosquito research field. For example, we've used it to study the functions of genes during development of the olfactory system, brain, midgut, and other tissues of vector importance.
The main advantages of this technique are that it is high throughput, relatively inexpensive, and less stressful to the organism than delivery of interfering RNA through microinjection, which requires a greater level of skill and cannot be implemented in the field.
Along with postdoctoral fellow Keshava Mysore, technician Ping Li will be demonstrating the procedure.
To begin, dissolve Chitosan in 0.1 molar sodium acetate buffer to make a 0.02% working solution. Keep the solution at room temperature before use. Then, dissolve double-stranded RNA or small interference.